So, what you're about to watch today is not science fiction. A few years ago, it would have been science fiction, but now it's reality. And thanks for tuning in to episode number 65, a new episode of the Doctor Podcast Show. And I'm your host, Dr. Robert Seichert. You'll really enjoy this fascinating episode, and we appreciate if you subscribe and follow us and like this episode and repost it as well. Today, we have a great guest coming from Switzerland, Dr. Fred Jordan. Dr. Jordan is the CEO and co-founder of Final Spark, which we'll talk about. He's got a Ph.D. in signal processing from EPFL, which is part of the Swiss Federation Institutes of Technology, very prestigious organization in Switzerland. Dr. Jordan is an expert in wetware computing and biocomputing, two new terms that you may have not heard about, but now you'll become an expert in them. So thanks very much for joining us today, taking time to join us on the Doctor Podcast Show, Dr. Jordan. We really appreciate it.
Well, thanks for inviting me. It's a pleasure to talk about this fascinating field.
Yes, it is. So tell us what a human brain organoid is. I know most people out there have never heard of this before, but it's something that's becoming very important and popular in today's age of brain-computer interfaces. So what is a human brain organoid?
Well, so human brain organoids falls into the general set of organoids and human organoids, okay? And so nowadays, there are a lot of companies working on specific organoids or some section of your body, let's say, okay? And so it started to appear, I would say, fundamentally, it was like Yamanaka in 2005 in Japan, Nobel Prize, invented a way to turn a differentiated cell of your skin into a stem cell. Okay, and from this, it was called induced pluripotent stem cells. It was possible for the first time to create differentiated cells of any part of your body. And one part of the body is the brain.
Right.
So
a very important part.
Yeah. And so what we do here at Final Spark is that we are differentiating cells from stem cells to glial cells and neurons, which are the two main cells that you will find in a brain. And we connect them in order to create a small structure that is called organoid.
So it's kind of a mini brain made from cells derived from stem cells that you somehow stimulate to produce neurons and glial cells, which are two of the predominant cells in brain tissue. Now, where do you keep these cells or organoids? Are they in test tubes or Petri dishes?
So there are different stages, okay? The stem cells themselves, we keep them in liquid nitrogen. Okay. And when we want to have more cells, so we thaw them, and there are specific protocols in order to do what is called an expansion. That means just multiply the cells. And from there, we are going to, we'll say, create the organoids, which are going to be floating in a liquid medium, like a small so-called well of like 6 centimeters of diameter. And we have medium into this, which is basically water with glucoses and vitamins and amino acids and a number of things. And they will be floating here, and the whole thing is going to be a so-called orbital shaker. That means it's going to move around in an incubator 24/7, and they will live there. And each small organoid is about 500 micrometer of diameter. So it's something that you can see with the naked eye.
So it's about a half a millimeter in size, you're
Yes,
saying?
yes.
Okay, so that's something that you can see with the naked eye. Now, why did you pick the specific cells that you chose? As you know, the brain has all sorts of different cells. They're all specialized in doing different things. Which specific cells did you pick to grow? Are these the neurons in the cortex? Is it in the hippocampus, the amygdala, or other parts of the brain?
Yes, that's a very good question indeed. Since we start from the stem cell, we have the choice, actually. And depending on the molecules that we are going to put at the moment of differentiation, we are going to guide the differentiation. So right now we do forebrain organoids, we do hippocampal organoids, and striatum organoids. So we have different types of organoids that we can even connect together.
You can actually connect them, which is
Yes.
amazing, and we'll get to in a moment. Now, in this half millimeter organoid or mini brain, I'll call it, approximately how many brain cells are in there?
About 10,000 neurons.
10,000.
Yeah.
Okay. And they bathe in a special liquid to keep them happy and alive and functioning.
Yeah, it's so-called Dulbeco medium. It was invented in the 60s, so this is not science fiction at all. It's a very
Right.
well known. We fall down in the field of cell culture, where protocols have been very well defined by the people before us. Where we are just reusing things which are very well known, because these are the things which work.
Right. Now, how do you prevent these mini brains from getting bigger, growing to 5 millimeters or 10 millimeters,
Yeah.
or growing into a full-size human brain, possibly?
Yeah, well, no, yeah. So there are two different questions here, okay? Preventing them from getting bigger and preventing them from becoming a normal human brain, okay? So the second thing is not going to happen, just impossible, okay? With the type of protocols that we have, we are always going to differentiate glial cells and neurons for a specific region of the brain. We would have to have a totally different protocol, and I don't I'm not aware of anybody who is able to do this, because then you have to grow and change actually the protocol in 3 dimensions as the system is growing. So okay, this is more than science fiction, I would say. So
Right, but when I interview you in 3 or 4 years, you'll be telling me how you grew different cells and connected them together. So right now it is science fiction, but things are progressing rapidly. Yeah, so go ahead.
so now, in order to stop the growth, so the first reason, the first thing to say is why we want to have small ones. Okay, we want to have small ones because big ones are dying. So I explain. Okay, at the center, we don't have any vascularization system, so we get necrosis at the center of organoids.
Right, so there's no blood supply to these cells.
Yeah, but there is no nutrients, no amino acids, no vitamins, no salts, no glucose coming in the center. Okay, it's a very well-known facts for all the people doing organoids. Okay, so this is why we have to keep small. How do we keep small? So this is again comes to the protocol. So the protocol says, for instance, you are going to shake at 80 rounds per minute. You are going to put 1% of BDNF and GDNF at this stage, and you are going to put this for 2 weeks. And if you do this, as soon as you differentiate a cell, it stops being a stem cell by definition. And therefore, it cannot multiply again. At least for the neurons, they don't multiply once they are differentiated. So this is how we stop the growth.
I see. So you're able to control this very well. Now, approximately how many of these brain organoids or mini brains do you have in your lab running simultaneously or living simultaneously?
So you said living and running, and I'm going to make the difference actually because there are two different numbers.
Right.
So for the living ones, normally, routinely, we have a few thousands of them. Okay.
1,000.
Yeah. Consider that we can keep them more than 2 years, living like this in the orbital shaker. It's okay. Okay. So these are organoids that we see once a week. Okay. When we do medium change, that means we have to renew the medium, and that's all. There is no interaction with them. There are the other ones with which we make experiments. Okay. And normally there are typically 16 organoids which are live. That if you go on our website, actually, you will see those 16 ones. They are live 24/7. And this one, because they are connected to electrodes, and because they are not in an orbital shaker, they are not in so good conditions, environmental conditions, that will not live as long.
I see. So you actually have tiny electrodes, wires connected into these brain organoids?
Yes, absolutely. So for each organoid, we are going to have 8 electrodes, which can be used in input and output. That means we can listen to the electrical activity, which in our case, we are going to listen to every individual action potential of the neuron. So really the full action potential, okay, in real time, sampled at 30 kilohertz. And we are going to be able also to stimulate by sending some very small pulse of current, and to which, if they want, they will reply.
Very interesting. So basically, you communicate with these tiny mini brains using electrical wires or electrodes, and you can send messages to them and also receive messages through them because all the cells in the brain actually communicate with each other via microscopic electric currents. So you're just kind of duplicating that, but you're connecting hardware into brain cells, right?
Yes, yes, correct, yes.
Wow, that's amazing. So tell us now the next step. What is biocomputing or wetware computing? It's also known as. Can you explain that?
Well, actually, yes. Biocomputing is the only thing which is new here, okay? Because making electrophysiology, the field of interacting with neurons, is known for 30 years. It's a very well-known field, okay? Which is wonderful because we have a lot of literature to inspire us, okay?
Right.
The thing which is new is to say, well, this neuron is a machine and I can use it for computation. That's all.
What kind of computation are you referring to?
So at this point, it's not very interesting computation, you know. We're happy when we can make an AND or OR gate, okay? But ultimately, there is, conceptually, okay, there is no barrier that would prevent us to make image recognition and more generally, for instance, any task which is currently done by artificial neural networks.
Wow. So potentially you could communicate with these cells, send them messages to do certain things. I'm exaggerating now, but can you tell the cells a joke, for example? And will they respond with some sort of electronic signal like, oh, that was very funny? It's a bit of an exaggeration, but what kind of interaction is there with these brain organoids?
Well, this is fundamental research, okay? So we are not at the joke level yet, okay? Uh, so if we send a stimulation, sometimes we will get in response some actual potential, some spikes, and sometimes not. Okay. What we are working on is to change the behavior of the network of neurons so that we get the answer that we want. And consider that everything that does AI is exactly the same problem that we have us. There is no difference in the problem statement. Okay, in AI, for instance, you present a dog or a cat to a system, and it has to tell you whether it's a dog or a cat. So there is like 10,000 neurons in input and 2 neurons output, dog, cat. That's it. This is how it works. AI, now it's artificial neural networks. This is very 101. What we are trying to do is to achieve exactly the same result. There is no difference. You still want to input some image, for instance, and get some output. We have some electrodes for inputting the image. We have some electrodes to get the output. So this is exactly the same problem. What is totally different is how you can achieve this. With artificial neural networks, so normal AI, you modify numbers. And these numbers represent the weight of the connections between neurons. So you're just manipulating numbers. Basically, you are more or less trying out some different numbers, so connecting the neurons differently in order that your output is dog or cat when it's a dog or cat. In our case, we cannot do this because we have no number, we have no access to the synaptic connections between each of the neurons. So we have to find another way. And this is the goal of the research, exactly this one. This is training of the neurons to do what we want.
So you're actually training human brain neurons as opposed to the AI networks, which are basically connected by NVIDIA chips. So that's the difference here?
Well, the fact that they are human is, at this point, I would say, from a functional perspective, a detail, okay? Because there is no obvious difference in the way a rat neurons is working compared to a human neuron. I would love to say that we are by far superior, but it's not obvious. So
Right.
the most important difference is that in one time you are using a simulation of a neuron, and the other time you are using the real neurons, a living neurons. And this is a view, a way to consider the problem which is new. And sometimes I say the only thing in innovation is to have a different view on the problematic. Okay, and this kind of thing saying, okay, I'm going to use the neurons and it's a machine and I can make something useful with it, is new. And the rest is engineering.
I see. One of the things I was fascinated by going to your Final Spark website is the electrical consumption difference between the AI networks that are now being created by the various AI companies as compared to the amount of electrical energy needed to run your human brain organoids. Can you explain the difference in energy consumption?
Well, I can explain the difference. I can characterize or quantify the difference, okay? But I'm not sure I can really explain why it's fundamentally different, okay? We are talking about a factor of about 1 million, okay?
1 million.
1 million. This is absolutely huge, okay? And this is actually the reason why we started to work on biology. Because, as you said earlier, my PhD is in signal processing. It's not at all in biology. Okay, we just happen to be a number of 3 engineers working on this problem with artificial neural networks, and we realized that if we want to have a bit realistic models, artificial ones, it's going to consume so much energy that it's totally unrealistic. So this is why we decided to switch to biology. But it was absolutely not natural move, you know, because we had to learn basically everything from scratch because we did not know anything about biology at the beginning.
So how much energy does a human brain consume per hour or per day? And then compare that to the huge AI neural networks that are being created by, let's say, Elon Musk that require massive amounts of electrical power. and yet they're still not as good as the brain, which is just a few pounds in the skull and uses much less energy. Why is that?
So I think here it's very good to have the order of magnitudes in mind, okay? So your brain, let's say 100 billion of neurons, 10,000 synapses per neuron, it's consuming 20 watts, about 20 watts, okay? If I wanted to make a very rough simulation of your brain, real time, okay, with the neurons totally unrealistic, very simplistic neurons, okay, it would consume already gigawatts of power. Actually, you would start to think about a small nuclear plant for one
Really?
brain. So you're talking small, nuclear plant compared to 20 watts. I mean, that's incredible. The gap is incredible.
Right. So if we could figure out how to design these human brain organoids and connect them properly, we could save a huge amount of energy and power, and we wouldn't need nuclear power plants to run these AI networks.
Yeah, and we will not need the future nuclear power plants that we are going to build if we go on like this, okay? And because ultimately our first goal is to commercialize cloud computing based on bioservers.
Hmm.
So I mean, one of the vision that we have is that we would like that people can still use AI, and we want this to keep on growing. It's okay, okay? But we want to be sure that there is no environmental impact of this growth of the use of AI.
Right.
And ideally, we should be in a situation running bioservers 1 million times, let's say, more efficient, that you come to a point where you can run from ChatGPT as you want. It doesn't matter anymore. The impact is negligible. A bit like light, there was light bulb before. This was a problem, lighting, okay? Now you have LED, the problem is gone.
Right.
Okay. So let's do it this way. Let's solve problems. So here there is only two ways. Either you find a nice way of producing energy without impact. People are still looking for this. There is still no solution. It may come. Or you find a way to do the same thing in a way which has less impact.
Hmm. Pretty amazing. What about, are there any ethical considerations? Has there been thought about whether there's consciousness in these brain organoids? Are we treating them right? It's potential moral and ethical issues. Have you considered that?
Yes, we take this very seriously. At this stage where we are now, we have about the same number of neurons than in the larva of a Drosophila, of a fly, if you want, okay?
Okay.
So, I'm all for the larva of Drosophila and I respect them, but normally we don't attribute a lot of consciousness to larvae of flies. But the question becomes interesting if we project in the future. Because as you said earlier, we can scale, theoretically, okay? If we wouldn't have this vascularization problem, which creates necrosis at the center, we could build 1 billion, 100 billions, or billions of billions, actually, of neurons. We have to stop them to multiply because it's free. I mean, virtually free. I mean, the cells just multiply hours after hours, okay?
Right.
So, and then you start to ask the question, wait a minute. You are creating nervous tissue with 10,000 neurons. Okay. But now if we have 10 millions of billions of neurons in your nervous tissue, what do you have actually? And the answer is, I don't know. Okay. And I'm afraid nobody knows. So it's the right time, I think, to discuss and to discuss with people who are ethicians, who are philosophers, who have been thinking and who are able to have the culture to think about these problems. This is not my field of expertise. However, I have one part of the field of expertise is just the technical part. And we are currently interfacing with a number of academic researchers in the field of philosophy to bring our knowledge and to say, what do you think about this? How do we project in the future? What does it learn? Does it teach us about ourselves, actually? So these kind of thoughts, I think it's appropriate to have them right now. So this is what we just started a few weeks ago.
Right. It's kind of the same dilemma with the silicon AI networks. You know, when do they achieve consciousness? And it's a problem that hasn't been solved yet. And it will take some time, and hopefully we won't make any big mistakes on the way there.
Yes, yeah. It's interesting because with silicons, you're right, with AI, people are asking these questions, but the questions seems way more hot when you start to use living human neurons. Okay,
Right,
so it's even and this is the, I think it has always been this way. When technology moves forward, it's ask us new questions, and
right,
we have to answer to this. And what is interesting is you can say, well, at the end, what is the purpose of technology? Is it good at the end? What I'm always considering is that I'm better living today than 1,000 years ago. And I think
right.
it's the case for many people. So I think the end result is good. It doesn't mean that everything is good. So this is why we have to take things cautiously.
Right. I think that's a great analogy, and that's what we want. Are we doing better now than we did before without endangering ourselves? Um, One thing that I found incredibly fascinating on your website as well is your neuro platform project, which allows people from outside of your labs to connect to the human brain organoids that you have there. Can you tell us about that, how that works, and what is the goal there?
The neural platform is an incredible story, I tell you, okay?
Yeah,
Because it was not in our plan to do this, okay? What happened is that the COVID came. And it's because of the COVID that we had a problem because we could not come to the lab,
right,
but we still wanted to do experiments. So what we started to do is to put some interface, electronic interface through the internet to everything that we do normally in the lab. That means stimulating the neurons, reading the neurons, but changing the pumps, putting some UV, a lot of things actually. And then we said to ourselves, well, actually, we are going to use Python interface, actually a programming language, to activate all this. So it's entirely automatic, wonderful. And then the COVID ended. But what we had actually is something that we could operate 100% from home, from
hmm,
30 kilometers away. And then, okay, we started to discuss with some universities. Are you interested actually to operate this? You are 5,000 kilometers away, but maybe you want to play with the neurons and make some experiments. And we have 34 universities. We said, yeah, we want it. This was
yeah,
not the plan of initial. And so we could not do this because the idea was to make it available for free. So we made it available for 9 universities. There is one in the US, which is Michigan, for instance. We have several in UK, in France, in Germany. So,
all
and
over the world.
yeah, it's all over the world. So the neurons are available 24/7, and everyone is booking, some time to do some experiments. It's really an open innovation program. So what we say is that if you want to work on the problem, which is biocomputing, that is training the neurons to do something, then let's do it together. We are going to this access for free. So we give this all for free. If people have good projects, we select the projects. We have so many, and
Right.
from time to time, so we give also technical support. So and we make sure that the neurons are happy, so they can make good experiments. And then we discuss from time to time about our results, their results, and we are moving this way. So that was what was published 3 months ago in Frontiers. So in Frontiers, we described the neural platform. Okay. what we did not expect neither is that some people came to us and started to say, well, we want to have the neurons only for us, and we don't want to share the results with you.
Interesting.
And we said, okay, why not? If you're ready to pay this time, okay? And they said, no, no problem, we can pay. Are you ready monthly? Yes, no problem. I will pay per month. Okay, so we started to rent to a first big company in Japan. Okay, and then to a second one in Europe, and there is a third one which is going to sign, I think, this week. So, and I think this one is a university actually that wants to use it for teaching. So because it's a wonderful platform for students in neurobiology, because they can really interact with real neurons and make some experiments and really get a feeling of what it's like. And this enables to have this into the master program without investing with all what we did.
Right. Yeah, it's a major accomplishment and setup in your lab. Can you give an example of what types of research some of the companies or universities are doing with your human brain organoids? Just
Yes.
an idea.
Yeah. For instance, there is something which is called the connectome. This is how the neurons are connected. That's it. Okay. So remember that when we put all these neurons together, they create a ball, but they connect randomly. Okay.
Hmm.
So the first thing that we have to do when we put the brain organoids on electrodes is to find what are the connections. And there is some mathematical approach to understand the connectome, so how they are connected. So this is very, very heavy on the mathematical side. And we had someone working for 6 months on finding the connectomes and trying to modify it, because ultimately this is what we want to do. This is an example. And I guess this will be published maybe this year or beginning of next year.
Right, because what makes a brain is how the different neurons interact and connect with each other. So if
Yes,
you can
absolutely.
figure that out, that's really incredible and amazing. Now, as you know, Elon Musk has his Neuralink company where he's created a working computer brain interface. It's really incredible where people can use their thoughts to control computers and eventually robots. And now he's working on actually curing blindness by connecting video cameras into the brain, the occipital cortex, via Neuralink as well. Have you contacted him as well? It seems like a natural fit between Final Spark and what you're doing and what he's doing. Uh, I think there are potentially lots of synergies between the two of you.
Yes, well, actually, I would love to have a way to talk to him, but I don't know how to do this. So,
Oh, I'll
so
give you his email address later after the show.
okay, thank you. But you're right, because the commonalities is that we are both working with human neurons. The
Right,
diff and we all both have organoids, organs. Okay, so organoid is kind of small organ. Okay, but there are some structural similarities between both. One of the big difference is that I can experiment freely many ways to connect with the organoid, and he cannot do this because he's not a real human being. So if we
right.
want to test something like new kind of electrodes or way to stimulate, we can check if something is good for the neurons and then creates a good interface. And then once we have it, then it can be used by Neuralink. So this is a good example of in vitro versus in vivo move translation.
Right. What about the potential security issues of people now connecting into your organoid network and possibly stealing data or changing data? Is that a concern?
So honestly, we are very open on the data. Okay, at this point, I mean, we have about I think maybe one of the largest database in the work for standardized recordings, because it's 4 years, 24/7, 17 gigabytes of data, terabytes, sorry, which are available for free, basically. So there is not a lot to hide here. So I'm not sure there is a lot to steal, neither. It's still a field of research. This may change in the future, but at this point, yeah, it's very open, actually. So we know everything. Most of the things which are done actually by the researchers are going to be published. So this will be open anyway.
I see. But as this becomes more sophisticated and let's say people connect the human brain organoids to AI networks, there may be some issues. Are you looking into that, or it's
No, we
too
are not yet.
premature?
Yeah, I would say really it's premature. We are not looking yet into this. I would say it's a good problem to have. So when we get
Right.
to this point of security issues, I would be delighted because that means that we have solved the first big problem, which is training.
Right. Um, is it possible in the future that you could eventually download data or information into these organoid networks and use them as a storage medium?
So, there are two parts in your question, okay? So if you say you want to put data into the nervous tissue, this is the scenario that you described, correct?
Right.
So in this direction, if I use the word of transhumanist, I would more talk about. I mean, I don't know if it's upload or download. What is the appropriate term actually? So if you consider to use nervous tissue as a storage medium, I would be a bit skeptical in terms of performance compared to static RAM or dynamic RAM. Okay, we have to be very pragmatic here about the performance. Not sure we can beat that with nervous tissues. However, if we talk about integrating a lot of information, like from millions of sensors, in order to perform a decision, then nervous tissue may be appropriate because we are good at this. We are an example of working integration system. And then it may be interesting indeed to have the possibility you take a brain organoid, which is new, and you teach it quickly how to do something. So in some way, you upload some know-how in order to react to our environment in order to make a decision.
Right. Have you considered using your systems to possibly do some research into brain diseases and neurological disorders? Since you're learning how the neurons interact with each other, it may bring some new information or insights into diseases like Alzheimer's, other forms of dementia, or neurologic disorders.
Yes, yes, you're right. We thought about this. This is particularly appropriate because, you know, you consider that when we do a brain organoid, I could do tomorrow a brain organoid of your cells,
Right.
of your own cells. Okay, so actually, it's really precise medicine when we can take one human being with a disease and duplicate his neural cells and replicate the disease he has, and then test some solution on it. And one indication that we get with electrodes is the electrical activity and how it reacts to stimulation. So yes, absolutely, this is a very interesting direction. And as a startup, we always have to present to investors what are the different possibilities, and this is clearly one of them.
Right. One of my colleagues that I interviewed about a year ago, Dr. Herb Blackman, who's at the Einstein Medical Center here in New York, is actually also doing some research with human brain organoids to figure out what some diseases are caused by, mental, various types of mental disorders and psychiatric, psychologic disorders. So he's doing research on that. You seem much more advanced, and perhaps you can come up with some answers, which will be amazing.
No, I wouldn't pretend one single second that I'm more advanced. But what I can maybe do is actually offer one way to test some things. For instance, if he has because we start to have this kind of collaboration, we have some people who tell us, Fred, we don't want to use your organoid, nothing personal, but we have our own organoid that we want to test on your system. Okay. So we are going to receive, I guess, this month, some organoids which are made somewhere else, and that we will put on the electrodes. And then the people making the organoids, I think they are in Germany, they will see remotely what are these activity, and they can interact electrically and see how their own organoids react and change over time. So this is a new way actually also to practice some experiments. You don't have a lab, to have all the electrophysiology, the pumps, and all the infrastructure that we have. You want to test a hypothesis, then we can do this, including incorporating some specific molecules that could be drugged tomorrow into the medium to see if it cures a specific problem that was found.
How does someone send you an organoid? What's the method of transportation, and how do you keep those cells alive and happy in the process?
Yeah, that's a real question, because normally the exchange, the international exchange of cells is done using a so-called dry ice, okay, which is basically a CO2 frozen. Okay, we are not sure that we are going. So this is good for isolated cells. In our case, we have an organoid, which is way more than one cell. Okay, it's a complicated system, and we are a bit skeptical. Well, some people have done it with liquid nitrogen, but it's a dangerous operation. So I think we are going to use systems when we are staying at 37 degrees. Okay, so we have to have some system that keeps the cells at this temperature in liquid medium. It can vary from 37 to 30 degrees. It's okay. So we still have to find a way to keep this temperature.
Right.
But this is a non-problem, actually. It's a very non-problem for the exchange of organoids and cells. But since it's in Europe, it's okay. When we will have the same problem in the US, then we have to find another way. Or I don't know.
Right. Now you mentioned that you're a startup and you have investors. How do you attract investors and get money to get to the next stage and become even more sophisticated?
So yeah, the relationship with money and the finance pack is very interesting. First, this is I'm one of the two co-founders of this company, but I made another company before, 23 years ago, which is called Alp Vision, which is doing something totally different, but is making quite some money. Okay, and we own the other company also. So basically, we have the finance to finance Spark. Okay, so that's the situation right now. But it doesn't mean that we don't need investors, because this is a new field. One investor was telling me last week that this is even a new industry, not simply a new field.
Right.
So, what we want to raise ultimately $50 million, starting with $10 million. And how are we reaching for investors? Well, we are not. Okay, we are not contacting investors. We just do not this because they contact us.
That's great.
So at this point, I was just looking at the Excel file this morning. We have now 18 investors who contacted us. Okay, we did nothing to get in touch with them. Mainly, it's except one. It's all from US or Asia. Also from Australia. So that's it. So nothing from Europe.
So
So
the
now,
fact that, yeah, go ahead.
yeah, please.
Yeah, so the fact that you're being contacted by investors is a proof of concept that this has huge potential. I think the pharmaceutical companies would have great interest in this because it would help them with drug development. As you mentioned, you can expose these organoids to various molecules and drugs and see what happens. So it might be a way to test pharmaceuticals even before they test them on animals, just to see what's going to happen.
Yes, sure. It's the thing with all startups, and this is what I saw with the first company, is where we have to be very flexible, okay, about what we imagine is the future from on the financial perspective and where the market is, okay. For instance, renting access to neural platform was certainly not on the radar maybe one year ago.
Right.
So, but we're also entrepreneurs. So if there is an opportunity, we're going to take it. And that's very interesting to see that there are not only VC who contact us, there are also people, individual people, consumers. Okay, and so these are very small investors. People write to us, I want to invest $1,000, $10,000 in, but these are not professional investors. Okay,
Right.
and so the first time we received one, said, okay, that's nice. I'm very happy to see that someone else in the world, as an individual, is ready to put his own money, because VC, this is not their own money. It's coming from funds. Okay, so it's very good, it's very big, but it's not a personal thing. And for me, it counted the first one who did that. But then there was a second one and a third one. And at this stage now, we have 450 people who contacted us and want to invest several thousands of dollars in our company. So at these kind of numbers, when you sum up the whole total of investment that would represent, this starts to be, it's called equity crowdfunding. So when you actually making a crowdfunding campaign and people, instead of receiving a product or anything else, they just receive some shares. Exactly like a public company, actually. So, but it's, IPO for the masses. I don't know. So, so we are trying to consider the different, what is the different possibilities, but for sure, we want to have investors who believe in what we are doing. And I'm so happy to see that there are many people actually who work for free for us. Okay. They just say, I just believe it is wonderful what you're doing. And I know how to do this. For instance, I know to make communication, social networks, or I know how to code in Python, C++, neurobiology, and they contact us and they work for us for free. Just
Really?
because
Wow,
they love
that's
it. Yeah,
awesome.
that's an incredible situation. So, so we have also to manage this mass of people who want to participate. So we are going to open also a forum, a Discord forum, when people can also discuss between us, actually, not only with us.
Right. That's incredible. Shows a great interest and huge potential. Where do you see Final Spark in 5 or 10 years? If you can predict that, I think right now it's still in its infancy, but has incredible potential.
Well, I was talking precisely about the future with a colleague just 2 hours ago, and there was this thing is that if more and more people, consider that we never marketed rental of the neural platform. Okay. We didn't make any ad, okay, and we already have customers.
Right.
This is interesting. Okay, what would it be if I start looking for customers? And and it's no problem. We can scale this up. Right now we have 5, 6 systems that we can run in parallel, but we could have 100, 10,000. It's okay. We are going to build the incubators and have the people work on it remotely. Wonderful. So this is one direction that could happen. Okay, but there's also the biocomputer side, pure biocomputer side. But I think the most important application of what we are doing, I'm going to tell you, I don't know. Because consider this. The first people who invented Shockley, for instance, he invented the transistor, okay, beginning of last century. I'm not sure he could guess that we would have smartphones and internet today running with, that's the thing. So this is the situation. So people ask me, what is the future? I don't know. I know what I have, but so we'll see.
Right. For a long time, transistors were just used to shrink the size of radios. So you could have
Yes, yeah,
a small transistor radio instead of a huge thing. And then
absolutely. Yeah.
things evolved. So I think that's a great analogy of where you're headed. So this has been a very fascinating educational conversation. I'm incredibly impressed with your company and what you're doing. I think it's going to be the future, and I think you're going to be contacted by lots more people and investors and people who want to work with you for free. That's the best sign of all. So I really appreciate you taking the time to join us today and explain what you're doing. Really appreciate it.
And thank you for your questions. I always love when I have informed questions that because it's always a pleasure actually to discuss about what I'm doing. So I was happy to be there.
Thank you.